Thank you, everyone, and thanks, Amber. I wanted to say what an honor and a privilege it is to be selected or asked to speak to you today about some of our work. By way of introduction, I'm an instructor and practicing adult epileptologist at MGH, and also a physician scientist doing a lot of work closely with Ann Paduri at Boston Children's, which is where much of my research is based. But all of this work has been independently funded as part of a KO8. And so without further ado, I'd like to talk to you about my approach to high throughput imaging of GABA fluorescence as a functional assay for GAT1 variants. Just as an overview to connect this work with my overarching interests, I'm often asked as an adult epileptologist, I should say I'm often confused for a pediatric neurologist due to the fact that I'm interested in genetics. And I don't see that as being too far from relevant to my work, because I'm interested in the molecular mechanisms of epilepsy, and specifically mechanisms related to epilepsy severity. And so this has effects on genetic generalized epilepsy. I should have also included developmental epileptic encephalopathies, but also in focal acquired epilepsy. So in the types of patients that I see most routinely in my practice, these patients might have a focal cortical dysplasia or stroke, but there's no routinely assessed clinical variable that seems to account for why we have some patients who have very refractory epilepsy and others with the same insult who do not, or have very easily controlled epilepsy. And so I'd like to thank Dr. Rogowski for his talk earlier, because many of the ideas he expressed resonate with my own, and that's that genetics really always matters. And in cases where there isn't a monogenic variant of large pathogenic risk, it's still likely the case that there could be, in aggregate, many small variants of low pathogenic risk, which together influences people's severity of epilepsy. And so that's a great interest of mine. But in addition to what Dr. Rogowski had mentioned, is that I really think that that's a powerful lens through which to approach the identification of new anti-seizure targets. And so thinking about genetics as a way of finding new target diversity for anti-seizure drugs is a special interest of mine. And so in addition to that, one of the things that interests me is developing tools and methodologies for accelerating basic and translational investigations. And so that includes cell-based models of epilepsy genes, also animal models of chemical and genetic seizures using mouse and zebrafish, but also methods for high-throughput chemical and genetic screens. So the work today really kind of addresses this sort of cross-section of my interests. So the objectives for the platform that I developed was to have a fluorescence-based assay for human GAT1 function using a genetically encoded IGABA sniffer protein. So, I have to give credit to the lab that developed this IGABA sniffer. This was not my invention. It was described in Marvins et al. From the Luger lab at Genelia Farms, very similar to a GCAMP. This is a protein that's a destabilized GFP that has an increase in its fluorescence in the setting of GABA, and so it can be used as an extracellular sensor of GABA or an intracellular GABA sensor. And this was a project to develop it as a cell-based assay for intracellular GABA for the use of testing human GAT1 function. And there have been radiometric assays for this. So again, it's not something that is a total groundbreaking thing, but by converting it to a fluorescent cell-based assay, there's certain advantages that can be taken of in terms of existing workflows that exist for high-throughput screening using high-content imaging. And so in addition to that, we wanted to assess the effects of human variants on GAT1 function using this assay, and then to optimize this assay for assigning likely pathogenicity to variants of uncertain significance, and also to optimize this assay for high-throughput screening for possible GAT1 agonists towards the question of trying to treat a condition that has haploinsufficiency as a mechanism by boosting the function of an existing wild-type copy. In overview, I think that we are making excellent progress towards this goal. For the nature of the constructs that we developed were that we have a construct that expresses HGAT1 and it's connected by P2A with a TD tomato fluorophore. And then in trans, we have an IGABA sniffer that has been additionally modified based on the reports from Marvin et al. And then in addition, it's been modified again to be intracellularly localized instead of surface expressed. And this is linked by P2A to an MBFP. And the idea for this assay was that all the cells would be triply labeled, but then in the presence of GABA, is there a mouse? No. In the presence of GABA, we would have increase in the intracellular GABA by way of HGAT1, and that this would increase the IGABA sniffer-related GABA fluorescence. And this is just an overview of the workflow. We've done this in HeLa cells, tried it in HEK cells, didn't work. It works by transient transfection and also in stable cell lines. It works in 96 well, 384. We use the Image Express Micro automated high-content confocal fluorescence imager and its downstream analysis pipelines. And from this, we're able to produce this estimate of GABA transporter activity based on the fractional increase in the IGABA fluorescence. Then this is just an example of what it looks like. This is an example from our stable cell line, which looks very pretty. And you can see in the vehicle-treated cells, they're all triply labeled, but just focusing on this one particular cell that's outlined with the dashed lines, you can see that the level of the IGABA sniffer-related green fluorescence is quite low compared to the level of fluorescence that you see after being treated with GABA at 100 micromolars. And then looking at the histogram of IGABA sniffer-related fluorescence across all cells in a particular site of a well, you can see that with 100 micromolar, there's a nice difference in the curves that allows separation of GABA. But then in the lower panel, I show you the results from having analyzed the average or median values from multiple wells within multiple, or I should say multiple sites per well across multiple wells. And so this is the metric that we're using for some of the next slides that we'll show. And so to get a ground truth for the assay, we wanted to know what GAT1 loss of function should look like. And before we got into any human variants, we used some deletion constructs to make that assessment. So at the top of the slide, you can see the protein architecture for GAT1. It has a cytoplasmic domain, an extracellular domain, and it has 12 transmembrane domains. And we used this delta CPD, delta ECD, and then a deletion construct that deleted the 6th and 7th transmembrane domains. And these had separately been shown to be deletions that were sufficient to abrogate the transport activity of GAT1. And here we ran a GABA series from 0 across 5 log fold. And you can see several things from this slide. First of all, there is a very nice increase in GABA. This is the IGABA metric that I'd mentioned. There's a very nice increase in this across the dosage range. That is also true for these deletion constructs, but it's really only at the high concentrations of GABA that becomes true. This would speak to there being some sort of non-specific uptake of GABA in these cells, but that these high affinity uptake of GABA is really restricted to these low concentrations. So GABA 1 micromolar, for example, there's essentially no appreciable uptake in any of these deletion constructs, whereas there's some non-specific uptake that starts to occur at higher concentrations. So it seemed like 1 micromolar was a good screening condition for variants that I'll discuss in later slides. And this is just to show the sort of test-retest stability in the transfection, a transient transfection approach. Given the variability of an assay that's based on transient transfection, we wanted to know what that would look like. And sure enough, when coefficients of variation in these group-wise averages are high, naturally this testing strategy doesn't work well. So being mindful of limiting these assessments to conditions in which the coefficients of variation are the lower the better gives you greater confidence. And even allowing replicates in which the coefficient of variation was somewhat high, like 0.05, but eliminating those that were greater than 0.10, we do get fairly good quality measures for the magnitude of the effect using robust, strictly standardized mean difference, as well as the robust C prime, which are measures that are often used in high throughput cell-based screens. So then we went further to ask how this assay would perform in the setting of known human variants, and we biased we'd like to do all of them, but at the moment, the preliminary data we have here, we started by biasing our list towards the most recurrent variants, so ones that had 12, 8, into ones that were, in some cases, only having been described once. And this list here is comprises 50 out of 88 cases that were reported at the time amongst missense variants in the SLC 681 Connect and other references when we were starting this work. And so we have some benign or predicted to be benign, some that are pathogenic or predicted to be likely pathogenic. And then we had a number of different variants of uncertain significance seen in yellow here. And again, this is somewhat preliminary, but it does serve the point of demonstrating that we are seeing many of the things that we would expect to see with this assay, and that is that the test retest value of various mutants in this list seems to be pretty high. In blue, we have wild type GAT1 and its performance based on this fractional increase in GABA metric. We have them across different plates, which is the reason for the different numbers. We have the deletion constructs in here, and then we also have the different variants, and they've been color-coded. And I'll show you another summary slide on the next slide. But this is, you can start to see, for example, that all of the known pathogenic variants in red appear to have values of fractional change in GABA that are well below that observed in the wild type. And then amongst the yellows, which are the variants of uncertain significance, there does seem to be a spread with some that have values that are definitely close to wild type and others that are essentially no fractional increase, suggesting that they do have true reduction in the GABA transport. And this is that data having been sort of summarized together, and then having applied a threshold of like 0.15 between these groups, you can see that there are major clusters related to VUSs that may have no strong reduction. They may still have a reduction, but just in this preliminary data, they do not seem to be as strong as some others that are indistinguishable from the level of GABA transport that's measured for some known pathogenic variants. And so just to summarize this, amongst the ones that we've tested so far, 6 of the 6 known pathogenic ones had a reduction. 5 of the 9 VUSs had reduced GABA uptake, whereas 4 of the 9 had no change. And then amongst the benign, 1 of them showed no change, and the other one failed our QC metrics that I had discussed. So in summary, this cell-based IGABA sniffer assay for GAT1 function is a non-radioactive method based on automated high-throughput imaging/analysis. It seems to be suitable for assessing deficits in whole cell GABA uptake based on fractional change in intracellular GABA. It does not assess surface expression, but it could with some modification. The original version of this assay was not linked. The HGAT1 was linked directly to TD tomato, and some initial data suggested that the values were indistinguishable. So it suggests that we could label it and it wouldn't affect actual GAT1 transport activity. So if that's of interest, then it could be done. And then you could have a single assay that is able to assess multiple mechanisms of potential pathogenesis. But there is something to be said for the fact that reduced surface expression is likely mediating its pathogenicity through a reduction in the whole cell GABA uptake. So depending on what the level of interest is, that is a potential direction. And it does seem to be suitable for assessing the pathogenicity of coding variants in SLC61. So in terms of next steps, we'd like to make this more robust and really establish the criteria for calling pathogenicity using some of these benchmarks. That might make this criteria something that could be more portable and be done by other labs. We'd also like to establish the criteria for enhanced GABA uptake to be able to call compounds in a high throughput compound screen as agonists of GAT1 activity or not. We'd also like to test variant-specific effects of GAT1 gene delivery, that is to say, dominant negative effects, just as a proving ground. It's certainly a question for maybe novel alleles that are discovered, whether or not they have a dominant negative effect, and this could be a proving ground for that when trying to decide a rational therapy. And then also high throughput compound screening, both for drug repurposing and also in collaboration with Collaborations Pharmaceuticals, we'd like to develop some GAT1 agonists through some generative AI approach. And so just to thank the people that have worked with me in my group, Guoqi Zhang did much of the work with his hands, and then it was my analysis, and then also the lab of Ann Paduri, and my funding source for this project, which was a K08.